Welcome to Chapter 11, Part 1, Mendel and the Gene Idea for EFB103. So this is all about genetics, and we're going to start off by looking at some of the early ideas of how heredity worked. Now, just intuitively looking around at living things, it doesn't take much to recognize that offspring tend to look like their parents. Maybe not completely identical, but they maybe appear to be a blend of the different parental characteristics. And so the early thinking on heredity had a lot to do with blending. And you can see some examples that might look like this. So we take, in this case, this black furred rabbit with a white furred rabbit. And the first cross here gives an intermediate characteristic. So we do see that sometimes. And we could even, in some cases, dilute that character further. And so there are some examples of where this happens, but it doesn't always work that way. Let's take an example here. So we've got a parental red flowered plant with these narrow petals, white flowered plant here with more broad petals. For example, the leaves on the stems are different numbers. All right, the first hybrid generation, when we cross these, we do tend to get like something that looks like a blend between these two parental flower species, plant species. But then when we cross these hybrids, this first hybrid generation, we get something that looks different. Yes, we see some of these hybrid characteristics, but the parental characteristics reemerge here, interestingly enough. So in comes Gregor Mendel. Now he's considered the father of genetics, and that doesn't mean that other people weren't working on it, but he really brought together a couple of areas that he was familiar with, which were botany and mathematics. You can look up his bio on Wikipedia. He had an interesting life. Check that out if you're curious. But the main point to take here is he brought in this idea of particulate inheritance, or the transmission of these factors that he called heritable units, rather than thinking about blending. So he studied pea plants, and here's a great example of using a model organism to test something in biology. He chose pea plants because you could make hybrids, so you could intentionally cross different types of them. There were a lot of varieties available. They're small and easy to grow, and this is really key. He could use self-fertilization or cross-fertilization to maintain or cross different lines of these pea plants. So this was a very useful setup. So here we have pea characteristics, such as, say, flower color here, or pea color, or pea shape, or pod color, pod shape, all these different things we'll get to. And these are called traits. So these character variants. So the character, say, is flower color, and then the trait is, say, purple versus white. Now, the other beautiful part of this is Mendel compared either or types of traits. So here the flower is either purple or white, not purple, yellow, blue, white. You start to add traits and things become more complicated. So how do we set this up? So here's a pea plant flower, and you have the male components of the plant and the female components of the plant. And so if you want to do a cross between two different plants, you can remove these anthers. This will prevent self-fertilization. And then you can bring in pollen from another plant. And so he would do this with just a brush, and people still do this today, crossing plants. Bring pollen from another plant. So here's flower from another plant. Bring the pollen over here, the mature pollen at this point. So you're removing those anthers before they mature. And then you can cross-fertilize. Another key point here is to prevent, say, insect fertilization or just random pollen floating around causing fertilization, is to cover that flower that you've set up in your experiment. And so this is also shown here. But I add the point to be made that you could self-fertilize. So use the pollen from the same plant. And this is very useful because that self-fertilization helps you, say, maintain a certain genetic line, which we'll see in a second. Or again, you could cross-fertilize with, here's pollen from this white flower. used to fertilize the this purple flowered plant over here. And as we'll see, if you were to do this, initially you'd get all purple flowered progeny from these seeds, these pea seeds. Another elegantly simple point here that Mendel used was the idea of starting with true breeding plants. What did that mean, true breeding? When he does those self-crosses, or anyone, if you do this self-cross on, say, a pea plant, you continually get the same characteristic showing up generation after generation. There's no hidden characteristic that reemerges like we see in some examples. So here we just see the same characteristic coming up time and again. That's true breeding. It's true breeding for this, say, flower color. Always going to get a purple flower color when we self this particular plant and its progeny. All right, so Mendel started with those true breeding plants. So here we'll start with the parent generation, so pea generation. And let's take flower color, for example. So he has true breeding purple plants, true breeding white plants, white flowered plants. He does this first cross, and this is the F1 generation, filial generation. That basically means son in Latin. It's think offspring. And this is the hybrid generation, but all the plants in this F1 had purple flowers. So the question is, well, where did the white go? What happened to those white flowered plants? And I will point out here, this is called a monohybrid cross. Mono means one. And so really we're looking at one characteristic. So flower color, for example, and we've got two traits, purple or white flowers. And so we're interested in one characteristic in a monohybrid cross. Officially, this step here, where we take these F1s, which are the hybrids from the parental generation, we're going to do another cross, or self or cross-pollination. This is technically that monohybrid cross. Now you got to set it up first by doing this first step, but this is the monohybrid cross itself. All right, so what happens in the next generation? The F2. Here that white flower color emerges again. So it didn't get just blended away. It didn't disappear. It reemerges in this F2 generation. All right, so what can be, at this point, what's the most significant conclusion that Mendel can draw from his experiments? All right, yeah, so he showed that blending isn't true. Now, as I showed you in an earlier example, it's not that people didn't really observe this before, but they were kind of confused by it. Everybody sort of felt, well, blending still sort of just makes intuitive sense, which is not very scientific. But nobody really looked specifically, like Mendel did, at these individual traits. All right, so he's looked at flower color now. And here it is, the flower color experiment. And I'll say these are just examples of some of his work. He did this over many years, but these illustrate the point. So he looked at other characteristics too, just again, one at a time. He's not trying to complicate things. He's just looking at one thing at a time. Yellow peas versus green peas does the same kind of cross, monohybrid cross. And what does he find in the F2 generation? All right, well, he counts up all these peas. You get a lot of peas out of these crosses. 6,022, 2001 green, and so on. And let's look at all these different characteristics. There's 7 of them. The thing to take from this is look at the ratio of one trait to the other. Purple flowers to white, 3.15 to 1. Here, yellow peas to green peas, 3.01 to 1. You see a pattern here. Notice a 3 to 1 ratio. Now, it's not exact because these are just, actual counts, real data. But there is a 3 to 1 ratio pattern here. So how did Mendel explain this? Well, he described the heritable factors that could account for this variation. We call them alleles now, thinking genetics. He didn't know any of that, but he had the idea right. So these factors, say for white flowers, it wasn't diluted or destroyed, but it reappears in that F2 generation, if we're thinking about flower, or one of these other characteristics, flower color, pea color, whatever it happens to be. With flower color, it's that white flower color that disappears in the F1 and reappears in the F2. Now, this would make sense if there are two different factors or alleles where one is dominant over the other. So the purple factor is dominant over the white factor. And then say that other factor, in this case, we're using white flower color. That is the recessive factor. So Mendel, again, didn't know anything about chromosomes or genetics. Nobody did at that time. But he had this general idea, right? If we look now, knowing what we know, we can look, say, at a pair of homologous chromosomes, and flower color is on those. This one chromosome has the allele or factor for white flower color. This other chromosome has the purple allele. So this is a heterozygote. They've got this individual plant has two different factors or two different alleles. And, we can look in more detail what we know now. Say this purple flowered allele produces an enzyme that allows the purple color to be produced. And if it's there, whether you've just got one or you had both purple alleles, you're going to get a purple flowered plant. So that's a dominant trait. So each organism inherits two alleles, and you could get a variety of combinations where an individual has two of the same allele. So that's these two examples here. That's considered homozygous. Homo means same. And you could have alleles being different. So this homolog has big A, this one has little a, and this is a heterozygous individual. Okay, all told then, Mendel came up with this idea of the law of segregation. Two alleles for a heritable characteristic. Again, he called them factors, but we're calling them alleles now. They segregate or separate during gamete formation and end up in different gametes. And therefore, a sperm or egg only gets one of the two alleles present in the organism. And this ties into the distribution of homologous chromosomes in gametes in meiosis, which we talked about in the last chapter. So here's a question for you. Which of the following phases of cell division confirms Mendel's observation of the segregation of alleles? All right, yeah, you have to think back a little bit to meiosis. It is in anaphase 1. Remember, that's when the homologs separate. The sister chromatids will separate in meiosis 2, but after meiosis 1, or specifically anaphase 1, the homologs have separated. So we see that here. The big A allele is going into this daughter cell. Little a alleles are going into this daughter cell. So this is a good time to introduce some useful genetic vocabulary that we'll hear time and again, really through the rest of the semester. One is the genotype. This is the set of alleles in an individual. So let's say we're talking about flower color. Big P, capital P. This would be a homozygous dominant plant. So this plant has that dominant allele. Capital P, lowercase p, this would be a heterozygote. Hetero means different. And because in this case, flower color is a dominant trait, this individual is still going to produce purple flowers. Heterozygote, as we'll see, we don't always have that dominant recessive relationship. We'll get to that. In this example, homozygous recessive, little p, this will produce that white flowered plant. Now, the other term that's important is the phenotype. This is the outward appearance of an individual. So the genotype, that's the underlying genetics. What happens in the phenotype certainly depends on the genotype to a large extent, but as we'll see later, there are other things that can influence the phenotype. Let's look back at this number, this 3-to-1 ratio, and what does this all mean? And can we use that in a way to predict what might happen if we're looking at new crosses or different situations? And so one way we can represent this is using something called a Punnett square. And if we think about what's happening during gamete formation, so let's take this parental generation, purple flowered plants, they're big P, they can only produce gametes that have that big P allele, right? That's the only kind of gamete you can get from this true breeding purple flower plant. Similarly, for this white flowered parent, only going to get that white allele, little p. In our first generation, F1, you have purple flower plants. That's the phenotype. These plants could produce big P or little p alleles. It's 50-50, right? Because some gametes are going to have the capital P, some will have the lowercase p alleles. And so now we have got a little bit of a different scenario. We can come up using this Punnett square with the possibilities. What are the potential outcomes? We're just connecting, bringing these gametes back together randomly. All right, big P could go with big P. So here's a possible outcome there. Big P could go with little p. This big P from the one parent can go with the little p from the other parent. And little p gives us our white flowered potential offspring here. So this is how we can do this. And again, let's just look at this simply thinking of the parental gamete types, sperm and egg, coming together in different combinations. Now, think of this from a probability standpoint. Sperm and eggs, we're talking about big R, little r now, same idea. There's a 50-50 chance that an individual is going to produce a big R gamete versus a little r gamete in their sperm. And then likewise for the egg over here. So there's a 50-50. So we can represent that by a half, right? So that's the proportion, 50% is half. It can be useful to use fractions instead of percentages because those will muddle how we look at this. All right, so let's put them together now. So, okay, well, big R could go with big R. That's a half times a half. That equals a quarter. Or big R could go with little r, etc. Another way to pick up on this is look at our square. We don't necessarily have to multiply half times a half to figure out that the likelihood of getting big R is 1/4. It is one block in this 4-part square, 1 out of 4. If we ask you here, what about the heterozygote? Well, there's a 1/4 chance of getting a heterozygote, but wait, there's another way to get a heterozygote. So it's a 1/4 plus a 1/4, or 2 out of 4 possibilities equals a half. So it's important to understand this just basic probability. When in doubt, you can draw out a square and see, just add up, count up the squares if necessary. But there are some shortcuts, as we'll see. So let's follow this along. So with Mendel's work, here's our parent generations, true breeding plants. Here's why that F1 is all purple, because the only possible gametes these can produce are little p up here or big P over here. Therefore, you're going to get all hybrids in the F1, but the hybrids could produce these different types in that F2 generation. So now that we see what's happening underneath the genotypic ratio, right? We have a phenotypic ratio, that 3:1, but we now recognize underneath what's happening is this genotypic ratio of a 1:2:1. One heterozygous dominant, one homozygous dominant, one homozygous recessive, and then 2 heterozygotes. Now, you could continue this on to do testing on this. You don't necessarily know which purple flowered plants in the phenotypically have the underlying genotype, homo or heterozygous. And so you could do selfing to determine whether they are homo or heterozygous. The white flower plants are always going to produce white. Only the homozygous dominant purple flower plants will always produce purple flower plants. Any heterozygote, you'll see those white flowers pop up again. So here we go. Here's that phenotypic versus genotypic ratio. This can become obvious when we look at other kinds of plants. So this is an example of snapdragons. These plants show a case of incomplete dominance. So it looks a little bit like the blending example. So here we have red flower plant. Here's the alleles CR and CR. That's how we annotate those for these guys. And CW for the white. These guys can only produce a specific one kind of a gamete. Here's our hybrid first generation with pink flowers. So here is a pink flowered hybrid generation. Self those or cross them with other hybrids. And here we see that 1 to 2 to 1 ratio. Let's talk about blood types now. This is always very interesting, and we're going to focus on the ABO blood groups. So this is going to follow the same Mendelian type inheritance pattern. In any individual can only have 2 alleles, but we actually have more than just 2 different blood types, as we see here. So blood type A, what does it mean when you're blood type A? That means your erythrocytes, your red blood cells, have this surface antigen, that's what we call it here. It's a protein on the surface that we've called type A protein. If you're blood type B, that means you have the B protein on the surface of your red blood cells. If you're AB, you have both. This is a case of codominance. And if you have blood type O, you don't have either of those, A or B. You don't have A or B proteins on your red blood cell surface. Now, this is important when we think of blood type matching, because if, say, your blood type A, you need to say a blood transfusion, you would want to get blood type A because your immune system is quite happy with blood type A. That's what it's used to. That's your blood type. But if you get, say, blood type B, your immune system recognizes that as foreign. And so you would develop anti-blood type B antibodies. Your body would react to it as something foreign. Let's use another example here. Your blood type O. You get any of these other blood types, your body would recognize them as foreign. But let's flip it around the other way. Let's say, let's say I'm blood type AB, and you get a transfusion with blood type O. That's okay, because your immune system isn't going to light up to something that's not there. There's none of those proteins on the surface. So you can receive blood type O. So what's happening at the genetic level? There are, again, 2 alleles, or each individual gets 2 alleles, but there are actually 3 different alleles in the population. So if you're blood type A, you might be IA, or IA little I. What's the little I? That's that absence of a surface protein. So if you have 2 little I alleles, you're blood type O. If you're type AB, that means you have an A allele and a B allele. And so this is how ABO blood type alleles are annotated. It's this IA, IB, or little I for that O type. And so you can work through exercise like this, just plug it back into the Punnett square like we've been talking about. So if we had a cross between 2 parents, blood type B and AB, we can segregate the alleles first into gametes. So here the male parent is going to produce IB or little I gametes. And the female parent here could produce IA or IB gametes. And then let's bring them back together. So what are the possible blood types for the offspring? Well, this is AB. Here's blood type B. This is also blood type B, but a different genotype version of it. And blood type A. You could ask, well, is it possible to have a type O offspring from this pairing? No, absolutely not. There's no way to get a little I offspring. All right, so as exemplified in this last row on the square, there's more than one way to get blood type B with these 2 different genotypes. And that's the same with our flower color example too, right? So sometimes we don't know the underlying genotype. So what can we do? And this works with flower color, not so much with human genetics, is to do what's called a test cross. So we could take our purple flower here, individual. We don't know whether it's homozygous or heterozygous, but we can cross it with a recessive plant. And we can predict, well, if our outcome is all purple flower, that means we must have had a homozygous plant. If it was split 50-50, purple and white, that means our test plant was heterozygous. And so depending on the outcome of your results, you can determine what you were dealing with. So what about two traits? Let's look at two things at a time now and see what patterns these follow. So Mendel did this with many different characteristics, and we'll just use the example of round yellow peas. These are dominant traits, roundness and yellow. So we can annotate those with big R, big Y, as our true breeding line of round yellow pea producing plants. Here's our recessive trait, true breeding plant. It's going to have the genotype little r, little r, little y, little y. We'll do a cross, and our first generation our hybrid. So this is the dihybrid cross of these individuals here in the F2. We would expect them to have big R, big Y, little y. So the question here, though, is, well, what happens next? So here's our parent, here's our hybrid offspring, here's the actual dihybrid cross. What happens? Is big Y always together with big R? So are they linked somehow so that the only possible gametes would be always big Y goes with big R, always little y goes with little r? And so that's the example here. So that we would expect a phenotypic ratio of 3:1. So we can set this up and do these crosses and figure out, does this happen? Or are these alleles independent of one another? So big Y could go with big R, or it could go with little r. And then little y goes with big R, little y goes with little r, and so on. And so we could make a Punnett square and connect our gametes all here and figure out what the ratio is. Ratio is this 9:3 to 3:1 ratio. Don't let this get sound like it's confusing. You're just counting squares here. There's 1, 2, 3, 4, 5, 6, 7, 8, 9 squares where we have a round yellow pea. All right, so Mendel did these crosses. Here are the data he got. Right away, you could see he was getting numbers or certain types that he wouldn't have expected if there was dependent assortment, that one allele always went with the other. Instead, he got results that supported this other idea that alleles for different characteristics are not connected. They are passed independently, so they assort independently. Now, Mendel didn't know about chromosomes, but we can look at this at a chromosomal level. Let's say here that one characteristic is on one kind of chromosome and the other is on a different chromosome. We see during meiosis that those are going to separate, so segregate. And this has to do with independent assortment, which we talked about when we talked about meiosis. And so you can get gametes that have these different combinations of alleles from different loci. And here's what this would look like. Again, if we're just overlaying the images of the chromosomes, noting that these different characteristics are coded for on different chromosomes. This is how this reflects what Mendel's results were. All right. So this is the law of independent assortment that Mendel came up with. So in a dihybrid cross, so when he's looking at two characteristics, each pair of alleles segregates independently. There's that independent assortment from other pairs during gamete formation. Now, there is a very important caveat here. Mendel, maybe through the wise choices of characteristics that he was looking at, or maybe with some luck with the characteristics he chose, that this law will only apply if the characteristics are on separate chromosomes. So that would be the example here. So here's gene A on this chromosome, pair of chromosomes, homologs. Here's gene B. They're on separate chromosomes. So you'll see independent assortment. But here, if we were looking at characteristics C and D, these are really close to each other on the same chromosome. And so they are going to tend to be passed together. This is called linkage. These genes are linked. So Mendel didn't have that situation, but we know this occurs, and that would certainly change the results if maybe by bad luck Mendel had picked characteristics that were on the same chromosome like this and close. So here we see independent assortment occurring during anaphase, really after metaphase. So it's that alignment of the chromosomes during metaphase that's going to, sort of shuffle them up. And then anaphase, that's where they ultimately separate. And that's going to result in different gametes with these different combinations given this scenario, where, say, the capital letter chromosomes or traits go north here, versus here, big A goes with little b. And so you end up with these different gamete combinations. All right, so let's think about different kinds of gametes that can be produced given a set of characteristics we're interested in. We're going to look now at 3 characteristics. So characteristic A, B, and C, and each one has 2 alleles. And in this individual with this genotype has 2 big A, so homozygous, but then heterozygous for the other 2 genes, loci. So what gametes can be produced? Well, I mean, you could just write out the possible combinations. So I call that the brute force method, right? Okay, big A could go with big B and big C. Then we'll take big A, big B, little c. And so you can write them all out. You would end up with 4 combinations, possible kinds of gametes. Or, this is more important, notice, well, the A gene here, that can only produce one type of allele. Or one type of gamete, I should say. The gametes are always going to contain big A allele. The B gene can give you big B or little b, so there's 2 common possibilities, 2 kinds of gametes, and then it's the same for the C gene. And so you can actually multiply these together. A, that's only going to give you one kind of gamete, so it's just 1 times. 2 times 2 equals 4. So basically 2 times 2 is 4. Another thing you could notice, which can be a shortcut when you get to bigger, more genes, examples of more genes, is that you can use this 2 to the n, where n is the number of heterozygous loci. So here we have 2. The B and C, those are heterozygous. So it's just 2 to the 2 squared equals 4. All right, you try it now with a bigger set of characteristics. How many unique gametes could you produce from an individual with this genotype? Okay, yes. So if you notice, there's only 3 heterozygous loci. And so that you could use that 2 to the n shortcut. So 2 to the 3rd equals 8. Let's look at blood type again. And some of you may have been thinking, well, I'm blood type AB negative or something. What's that negative mean? That is called the RH factor. And so this is another kind of protein that you would find on the surface of an erythrocyte. And so if you're RH positive, that means you have that protein on your red blood cells. If you're Rh negative, you do not have that protein. Simple as that. And it's a dominant recessive relationship. So if you have, we're going to use D as the big D, little d for the alleles for the Rh factor. If you have at least one big D allele, you're Rh positive. You'll have that protein on your surface of your red blood cells. Now we could look at these more complex crosses here. So here we're going to say parent number 1 here is B positive, and we're going to get we've got the genotype. They're IB little I, so they're heterozygous. They're also heterozygous for the Rh factor. And you could come up with the different gamete types. They're just noted up here in smaller letters. These are the possible gamete outcomes from this individual. Now, you can pause it here and ask yourself, well, what gametes could be produced by this AB positive individual, given the genotype here? And then if you wanted, you could plug these into a Punnett square, a 4x4 Punnett square, and think about what are all the possible offspring genotypes? What are their possible blood types? And there might be, as you'll see in a second, a reason why that Rh factor in particular is important to know about. So here's what blood type distributions look like here in the United States. These are data from the American Red Cross, divided by different groups of people. We see different proportions of blood types in different ethnic groups. And so you can get a sense of which blood types are more common or more rare in these different groups. And this is going to be different. Again, think of genetic background, depending on where you look in the world. So let's look at this RH factor again, this positive or negative. It's really important if, say, a mother is RH negative and there's potential that her offspring might be RH positive. So the male parent would be RH positive, so passing that RH allele to the offspring. Now, the reason for that is, say, during one pregnancy here, what we see is the mother will develop antibodies to this RH positive offspring. Usually that first baby is not at any risk of an immune response against it, but by the time, say, a second child is in utero and is potentially RH positive, the mother's immune system has been primed to this RH factor as something foreign. And so there's the threat of an immune response against that fetus. So there are treatments for this. It's important to know if you're RH negative, that this can be treated. So this is something that a physician might ask about if you're carrying a child and you're RH negative, that you might get this RhoGAM treatment or some other similar treatment to prevent that immune response from developing against that unborn fetus. Okay, let's crunch some numbers now before we move on to the next part of this chapter. You can start to see a simple Punnett square is a great way to solve some basic problems. But when you start to deal with more and more characters, it looks like things get more complex. But something that's super helpful here is to notice that even when we're dealing with a dihybrid cross in a multi-character situation or mini hybrid cross, you can separate out those individual monohybrid crosses and treat them as they're occurring simultaneously. Let me show you what that means. So let's take like Mendel's example here. We're using our dihybrid cross. So we've got our hybrid individuals in the F1 generation. And then we could ask, well, what's the probability of getting a big Y, big Y, big R offspring? Now you could make that big square and look back and count how many of the 16 squares have this outcome. Or you can look individually at, well, what would happen just with our Y alleles, again, in our parent generation here, the hybrids, and the R alleles. The probability of getting a big Y, well, that's a quarter. The probability of getting a big R, that's also a quarter. And so now we know the probability of those individual things, those individual outcomes. So we see here. And then we can multiply those together. So a quarter times a quarter is a 16th. If we go back to our big square, we would have had this outcome 1/16 of the time, but this is a much quicker, elegant way to do this. Now you try. So what's the probability of getting basically a heterozygous offspring for both of these genes? All right, so the first step here was noticing that there are two ways in these squares to get a heterozygote. And so the flat probability of getting a heterozygote is half. So probability here is half, probability here is half. And so we're multiplying half by a half or a quarter. Again, you could go back to your big square and just count them up, but this is much more elegant. How about this one? Let's take many more characteristics and different genotypes. Things look like they're getting confusing, right? But again, just look at them one at a time here. So in this example, we have this cross between this genotype individual and this genotype individual. And you could say, well, what's the probability of getting this outcome? Well, what's the probability of getting little p from this cross? Big P, big P by little p. That's just our classic monohybrid cross. So it's a quarter, and that's what's shown here. Little y, that's a half, because this individual is a heterozygote, but this one's always going to contribute a little y allele. So if, again, plug it back into a square, and you'll see 50% of the time, or half, you'd get little y, little y. And you can follow that same logic for any of these. So this is a nice shortcut to understand this. All right, so you try. Here's this cross between these two genetic individuals. What's the likelihood of producing this offspring genotype? All right, so here's that answer, 1 in 64. Again, you're just saying, what's the likelihood of getting big A from this heterozygous cross? It's 1 in 4. This is also 1 in 4, 1 in 4, so it's 1 over 4 cubed, or 1/4 cubed, gets you 1/64. So you can come up with all kinds of examples, try them out, but once you get that basic pattern, it's really fairly straightforward. So work through a couple of these, and we'll see you in the next video.